let's do it. So welcome everyone to this session of Let's Talk Flow. Thanks you guys for joining. Today we're focusing on the concept of unmixing. The main objective of this presentation is to quickly dive into two key concepts that will help you understand how to optimize your spectral experiment. So my goal is to refine our understanding of conventional versus spectral flow cytometry and ensure we're all on the same page when it comes to what unmixing really means. To quickly recap, last month we discussed why compensation is essential in conventional flow. And it's crucial to understand that on any instrument, every fluorophore is emitting light over a specific range of wavelengths that are unique to that molecule. So when you're running an experiment with two or more fluorophores, their emission ranges often overlap. For example, let's consider PE and Alexa fluorophore 88. If you're working on an instrument that only has a blue laser, Alexa fluor 488 is typically detected in the B1 detector, so the first detector off the blue, represented in the green shaded area, while PE would be detected in the B2 detector in the yellow shaded area. As you can see, there is spillover from AF488 into the PE channel. So what do we mean by spillover? We're talking about this emission overlap from one fluorophore that bleeds into the collection channel of another. And this is where compensation comes into play. Compensation is the mathematical method your cytometer is using to correct for this spillover. So let's highlight the key differences between conventional and spectral flow cytometry. In conventional flow cytometry, you typically have a one-to-one relationship. One fluorochrome corresponds to one channel. The number of detectors on your instrument determines the maximum number of parameters you can actually use in your panel. So in this type of setup, fluorophores are detected using a specific bandpass filter, and that filter is tied to a single channel. A laser excites the fluorophore and it emits light in a specific range, and it's captured by the assigned detector. Compensation is applied to correct for any spillover into neighboring channels. In spectral flow, an array of detectors are used that cover a broad spectrum. And here's the key difference. Every fluorochrome in your panel is detected across all the channels. So instead of a one-to-one relationship, the entire emission signature of the fluorophore is captured. These light signals are then pieced together from all the detectors, and they're deconvoluted during the unmixing process. And so to continue this comparison, at the top of this slide, this is an image of PE from our spectral viewer showing its excitation in the dotted line and emission wavelength, the shaded yellow. The vertical line that's kind of highlighted here is representing the bandpass filter that's capturing a specific range of that PE fluorescence. So that's the bandpass filter I was referring to in conventional flow, only focusing on that narrow range and isolating a portion of the emitted light. At the bottom, we see the spectral signature of PE. So in spectral flow, instead of isolating a single peak, we're capturing the entire emission profile across all detectors. The signature of PE is in more detail, and we can see in more detail the areas of emission. So spectral flow cytometry leverages information from all filters to create a comprehensive spectral signature. Both compensation and unmixing are techniques used to correct for overlapping signals from different fluorescent molecules, and both are handled by the software. But while compensation involves subtracting unwanted light from neighboring detectors, spectral unmixing is more like a deconvolution, and it's where there is a disentanglement of the contributions of each dye to the overall signal in your fully stained sample. So what exactly is a spectral signature? At the top of this slide, we have a BUV 737, and we're looking at a signature in a little bit more detail. So first, when you're looking at this, you can kind of think of it like a histogram turned on its side. The y-axis here represents the intensity of fluorescence ranging from 0 to 10 to the 4 on this type of scale. And in this example, we can see that the fluorescence intensity is increased in the last two detectors on the UV region. So as I said, this is the signature for BUV 737. So it makes sense that as the range for detection begins in the 300 nanometer range and ends in the 800 nanometer range. And so we have that 737 represented here. So now I would like to bring your attention to the bottom of the screen where you, I'm sorry, the bottom of the illustration where you see this reddish shaded area. And what this is the autofluorescence from the cells or the beads that were stained with BUV 737. So it's important to note that in a single color control, when it's actually run on the instrument, it contains both the fluorophore emission and the autofluorescence of whatever particle you're using. They're both present in the tube. You may also notice a bit of scattering here, like in some of these bars, which could also be maybe some carryover from other signatures or controls if you're running them back to back. So this is just to show you what a real life example of as you're running may look like. And this data is taken from a Bigfoot spectral cell sorter. So now when we look at the bottom, this is still BUV 737. However, the signature has been normalized. So before unmixing actually happens, normalization occurs, and it's defined as the process of subtracting this autofluorescence from cells or beads that have been stained and ensures that only the fluorophore signature is used for a reference to deconvolute the fluorophores in the sample. So a panel design tool will show a clean normalized signature, which means that the tool has already accounted for autofluorescence. A well-defined signature is really paramount in spectral flow cytometry. Poorly defined signatures can lead to errors in unmixing. And signatures can also help identify issues such as, malfunctioning lasers and detectors, contamination, and any problems with antibody performance. So here we have PE. It has its primary emission peak in the yellow laser, a secondary emission peak in the blue, and it also has tertiary emission peaks in the violet and UV lasers. So for instance, if you're experiencing issues, let's say with your blue laser, you'll notice that the secondary emission peak is for PE is missing. And so it'll offer some indication that something's off. You can also use the signature to troubleshoot contamination or tandem die breakdown. So let's say you're trying to look at PE size 7, but you notice that the PE signature is dominating. This might give an indication that there has been some significant tandem breakdown. So this is just an illustration that, paying attention to the signatures is crucial when you're running them on your flow cytometer. So to reiterate this importance of the reference signature in the process of unmixing, remember that the fully stained sample includes a combination of fluorophores, and it's the job of the algorithm to use these reference signatures as a guide to determine the contribution of that fluorescence in the fully stained sample. This process eventually gets you to your unmixed events on the 2D density plot. So the algorithm is using reference signatures provided through your single color controls. Remember that the first step in unmixing is normalization, and it involves the subtraction of the autofluorescence of that unstained particle, a cell or a bead. And this subtraction is crucial because it's how the software isolates that fluorophore signature. Here we have PEEF610 and FITSI, and these are the theoretical signatures. This is what the algorithm wants to grab. But remember on the previous slide when I was showing BUV 737, you have a combination, you had that red shaded area at the bottom, which is indicating the autofluorescence of whatever particle you're using. So here on this slide is kind of another illustration of the same thing that I've been saying. In the blue, we have the signature of the fluorophore. And in this example, cells were used as the reference control. So in the red, we have the unstained cell signature, or just the autofluorescence signature of the cells in the reference control. And so you can think of your actual reference control as two signatures in one. And so normalization, it involves taking that unstained particle signature and subtracting it from that control that you're using. And this in turn will give you the signature of the fluorophore. This is the information the algorithm is using to identify the fluorophore in the fully stained sample. So I want to iterate that the particle type you're using for your unmixing, it's not that these or cells are better than the other. It's just that if you're going to use one or the other, you have to make sure that the unstained is matching. You have to keep your controls consistent in that way. And so here's what happens if a mismatch between the unstained portion of the negative and the positive occurs. We start again with our reference control in blue. Then we have two negative unstained signatures. One is unstained beads, one is unstained cells. In this case, which is the correct one? Well, we started with a cells control that was stained. So we want to make sure that the unstained cells of this same control, same type of cells, are used so that when normalization occurs, we get the correct fluorophore signature. If this is not matched, then we see that the fluorophore signature will include some different emission profiles. And when the algorithm goes to find the contribution in your fully stained sample, that's why you will see unmixing errors because there is not a correct match. And so moving on to like our next point, first point being making sure the autofluorescence matches, we want to have optimized single color controls. And to no surprise, actually, the rules apply for both conventional and spectral flow. And so, just make sure your controls have these kinds of features. You want to make sure your positive and negative populations are clearly separated. The single color controls really need to be treated the same way as the test samples. And a perfect example of this is fixation. There are some exceptions to this. If you're using beads as your reference control, we do recommend to fix the beads, but some compensation beads will not allow fixation or not compatible with fixation. So that's why I recommend the ultra comp beads, because you are able to actually apply that buffer to them. It's because fixation does affect the fluorophore signatures. And the next point, you also want to make sure there are sufficient events in the positive peak. And this can vary from instrument to instrument. On the Bigfoot, for instance, there needs to at least be 400 events in that gate. And here is our fourth point, which was what I was previously talking about in the previous slides, is that the positive and negative events need to have identical autofluorescence. And this also can be something to think about, not just with the particle itself, bead and bead, cells and cells, but you can also think about the cell type that you're using. Like if you're using spleen cells in your reference control, make sure that the unstained cells are the same cell type. And also activation state. This can influence that normalization subtraction process. And so lastly, the fluorescence spectrum of the positives must be identical to the experimental sample. And you may have heard this before, you cannot interchange AF488 and FIT-C. And this is something I feel some may have gotten away with maybe on a conventional cytometer, but with Spectral, it becomes even more important to ensure that if you do have FIT-C in your sample, that you are using FIT-C in your reference control. So along with your controls, you want to adjust the selection of the positive in your positive peak. You want to adjust that for correct unmixing. So quality unmixing is also dependent on what the algorithm is using as the positive. So you can see here by grabbing the middle of the peak, we are looking at an unmixing error that has been translated into the actual data because this is not capable of grabbing this area of the peak is not possible to account for the brightness of these events here, which is why we see this curve here. But if you make sure you grab the tightest and brightest, you can ensure that the algorithm has the right MFI to account for all those cells that are bright here. So essentially errors could also be observed when the selection cannot or does not focus on the brightest population. And so essentially, understanding and correctly implementing spectral flow cytometry concepts is crucial for your data analysis. And I would say the 2 points that I want you to take away is that you need to prioritize well-defined signatures, ensuring that the autofluorescence of the positive and negative match. And secondly, ensure that the rules for optimal controls are followed and that for the positive peaks, you are getting the tightest and brightest. And both of these points, I say in summary, can be put in this main takeaway. Really want to make sure that the normalized signature matches the signature in the fully stained sample. And so hopefully with that, it will enable you to see less unmixing errors in the future.